Welcome to Chapter 2, the chemical level of organization. In this chapter, we'll take a step back from tissues and organs and zoom in on the molecular level to understand the chemistry that makes life possible. We'll explore the structure of atoms, how chemicals bond, and why reactions are central to everything in the body does, from building tissues to producing energy. We'll also examine the essential inorganic and organic compounds like water, carbohydrates, proteins, and nucleic acids. By the end of this chapter, you'll have a solid formation in biochemistry that will help you make sense of more complex physiological processes in later chapters. To truly understand anatomy and physiology, we have to start with chemistry. For every structure in the body, from a muscle fiber to a neuron, it's made up of atoms interacting through chemical bonds. The majority of elements, oxygen, carbon, hydrogen, and nitrogen, make up over 96% of our body's mass and are the building blocks of biomolecules like proteins and DNA. Chemical reactions are happening constantly to keep us alive, whether it's muscle contraction, nerve signaling, or breaking down food for energy. That's why this chapter is essential. It lays the groundwork for everything that follows. If we understand atoms, molecules, and how they interact, we'll be better prepared to understand cells, tissues, organs, and ultimately the entire body. Atoms are the smallest unit of matter that still retain the properties of an element. Each atom is made up of three types of subatomic particles. Protons and neutrons are housed in the dense nucleus, while electrons orbit around the regions called energy levels or shells. The number of protons defines what element it is, and that is also its atomic number. The mass number includes both protons and neutrons. If atoms gain or lose electrons, they become ions, which play key roles in electrical signaling and fluid balance. Isotopes are variations of elements with different neutron numbers, and some are radioactive, which can be useful in medicine. Knowing how atoms are structured helps us understand how they bond and interact with chemical reactions throughout the body. Atoms are the basic building blocks of all matter. They consist of three subatomic particles: protons, neutrons, and electrons. Protons and neutrons are packed tightly in the atomic nucleus, while electrons orbit a specific energy level or shell. Protons carry a positive charge, electrons are negative, and neutrons are neutral. In a stable atom, the number of protons equals the number of electrons, resulting in no net charge. Understanding these particles helps explain why atoms interact and form chemical bonds, which is the basis of all physiological processes. The atomic number of an element is determined by the number of protons in its nucleus. This number is unique to each element and is what defines it. For instance, all atoms with 6 protons are carbon. The mass number adds the number of protons and neutrons together, which, since those are the heavy components of the atom, electrons have very little mass and do not significantly contribute to it. When reading the specific periodic table, you can identify each element by the atomic number and predict its mass based on the average mass of its isotopes. Electrons don't just float around randomly around the nucleus, they occupy specific energy levels called shells. The first shell can only hold 2 electrons, the second shell up to 8, and so on. The outermost shell, known as the valence shell, is specifically and especially important. Atoms are most stable when the shell is full. If it is not, the atom will seek to gain, lose, or share electrons to achieve stability. This gives chemical bonding, which, through giving up electrons or forming ions, or sharing them, forming covalent bonds. So an atom's reactivity is all about its electron configuration. When atoms gain or lose electrons, they become ions. A cation has a positive charge due to losing electrons, while an anion has a negative charge from gaining them. These charged atoms are essential in physiology. Ions like sodium, potassium, and calcium play major roles in nerve impulses, muscle contraction, and maintaining hydration. Isotopes are variations of the same element that differ in neutron number. Most are stable, but some, like radioactive iodine or carbon-14, are unstable and decay over time. These radioactive isotopes can be useful in medicine for imaging or treatment. Chemical bonds are the forces that hold atoms together to form molecules. When atoms transfer electrons, they form ionic bonds. One becomes positively charged and the other negatively charged, and the attraction holds them together. This is a common in salt like sodium chloride. Covalent bonds, on the other hand, involve shared electrons. If the sharing is equal, the bond is nonpolar. If unequal, it becomes polar, like a water molecule. Hydrogen bonds are weaker but critically important. They hold water molecules together and stabilize DNA structure. The type of bond affects the molecule's shape and how it interacts with others. This directly influences biological functions like enzyme activity, hormone binding, and nutrient transport. Understanding bonds is key to understanding molecules' behavior in physiology. Ionic bonds are created when one atom gives up an electron and gains another. This electron transfer results in ions, which are charged atoms that attract one another. For example, sodium loses an electron and becomes positively charged, while chlorine gains an electron to become a negatively charged ion. These opposite charges pull the atom together to form a compound like sodium chloride or table salt. Ionic compounds readily dissolve in water and are essential to maintaining proper nerve function, muscle contraction, and fluid balance. In physiology, the flow of ions like sodium, potassium, and calcium across the membrane is key for cellular communication and energy use. Covalent bonds form when atoms share an electron. This type of bond is strong and common in biological systems. In a nonpolar covalent bond, electrons share equally. Think of molecules like oxygen gas or methane. In a polar covalent bond, one atom pulls the electron closer to itself, creating a slightly charge on either end. Water is the classic example. The water atom pulls electrons more strongly, making it slightly negative. While the hydrogen atoms are slightly positive, this polarity is essential because it allows water to dissolve many substances and form hydrogen bonds. Covalent bonds give structure to DNA, proteins, carbohydrates, and lipids, the molecules that make life possible. Hydrogen bonds are weaker than ionic or covalent bonds. They play a critical role in biology. They occur when a hydrogen atom that's already in a polar covalent bond is attracted to another nearby electronegative atom. In water, this creates a cohesion that allows for surface tension and capillary action. In DNA, hydrogen bonds hold the two strands together between base pairs. In proteins, they help maintain the molecule's 3D shape. Because they are easily broken and reformed, hydrogen bonds are perfect for dynamic processes like enzyme activity, cell signaling, and temperature regulation. Though weak alone, their collective strengths are powerful effects on biological systems. Chemical reactions are the basis of all body processes, from digestion to muscle contraction to DNA replication. In a reaction, substances called reactants are transformed into products. Synthesis reactions build larger molecules from smaller ones, like combining amino acids to make proteins. These are typically anabolic and require energy. Decomposition reactions, on the other hand, break large molecules down, as we see in digestion. These are catabolic and often release energy. Exchange reactions involve breaking and reforming bonds to rearrange components. Many biological reactions are reversible and can adjust depending on cellular conditions. Enzymes are vital here. They act as a catalyst, speeding up reactions by lowering the amount of energy needed to get started, which makes it life-sustaining processes efficient and timely. Synthesis reactions are essential for building the structures and molecules your body needs. These reactions take smaller units like atoms or smaller molecules and combine them into larger, more complex substances. For example, amino acids combine to form proteins, and glucose molecules link up to form glycogen for energy storage. These processes require energy and are often referred to as anabolic reactions. They play a key role in growth, tissue repair, and even hormone production. Without synthesis reactions, your bodies wouldn't be able to build muscle, form new cells, or store nutrients for later use. Decomposition reactions are the opposite of synthesis. They break down larger molecules into smaller, more usable pieces. They are critical for digestion and energy production. For example, breaking down carbohydrates into glucose or proteins into amino acids allows the body to absorb and use nutrients. Cellular respiration is another key example. Glucose is broken down to release ATP, the energy currency of the cell. These reactions usually release energy and are classified as catabolic. They are also important for eliminating damaged or unnecessary compounds, keeping cells efficient and functional. Exchange reactions combine aspects of both synthesis and decomposition. Bonds are broken and new bonds are formed as part of molecules are swapped. These are common in bodily processes that regulate pH or maintain homeostasis. For example, when an acid and a base combine to form water and salt, this is a classic exchange of reaction. Buffer systems in the blood, like a bicarbonate buffer, also rely on these reactions to quickly stabilize pH levels. They help the body adapt to changes without throwing off vital chemical balances. These reactions happen constantly, especially in the bloodstream and digestive tract. Not all reactions go in one direction. Many are reversible, meaning they can proceed forwards or backwards depending on the body's need. These reach a state called dynamic equilibrium, where the amount of reactants and the products stabilize. This flexibility is key to regulating metabolism and maintaining homeostasis. Enzymes play a key role here. They're biological catalysts that lower the activation energy required to start a reaction, making it faster and more efficient. Enzymes are highly specific to their substrates and are reused over and over again. Without enzymes, more bodily reactions would occur too slowly or to sustain life. Water is absolutely vital to life. It makes up about 70% of the body's weight and plays many roles. Its ability to dissolve substances makes it a perfect medium for transporting nutrients, gases, and waste products in the blood and interstitial fluid. Water's high heat capacity helps stabilize body temperature, preventing dangerous fluctuations. It also lubricates and cushions joints and organs, reducing friction and protecting tissues. Chemically, water is reactive. It participates in hydrolysis reactions that break down molecules apart and dehydration synthesis reactions that build them. Its polarity and hydrogen bonds make it possible. Without water, the body simply wouldn't carry out the chemical processes that sustain life. One of the water's most important roles is that it is a universal solvent. Because of its polarity, water can dissolve many substances, especially ionic compounds like salts and polar molecules like glucose. This property is what allows the blood and lymph to carry oxygen, nutrients, and hormones, as well as waste products throughout the body. It also helps cells take in what they need and expel what they don't need. Water forms hydration shells around dissolved ions, which make them more reactive and accessible for chemical reactions. Without this dissolving power, most reactions in the body simply could not happen. Water has a high heat capacity, which means it can absorb a lot of heat energy before its temperature rises. This property is vital in the human body. As metabolic reactions release heat, water buffers the system to keep our internal temperature within a narrow, safe range. Blood carries the heat throughout the body, while sweating releases excessive heat through evaporation. This thermal stability protects tissues from overheating during exercise or illness that helps maintain homeostasis in various environmental conditions. Water is essential for lubrication and cushioning throughout the body. Its major components of synovial fluid in joints helping reduce friction during movement. It's also found in the serous membranes, reducing friction between organs like the lungs and the heart. Water-based fluids like tears and mucus help surface moist and protected. Cerebrospinal fluid, or CSF, surrounds the brain and spinal cord, providing shock absorption and mechanical protection. Without water-based lubrication, tissues would wear down quickly and be vulnerable to damages from daily movement. Water isn't just in the background. It actively participates in many of the body's chemical reactions. In hydrolysis reactions, water is used to break down chemical bonds. This happens during digestion, where complex molecules like proteins and carbohydrates are broken down into smaller units. In dehydration synthesis, water is removed to help form new bonds, as when building muscle proteins or forming DNA. These processes are fundamental to help metabolism, your body's ability to break down nutrients for energy and build structures for growth and repair. Without water's chemical reactivity, none of this would be possible. Maintaining the right pH is critical for health. Acids increase the concentration of hydrogen ions in solution, while bases either release hydroxide ions or bind to free hydrogen ions, reducing their concentration. The pH scale runs from 0 to 14. Lower values are more acidic, higher values are more basic. Pure water is neutral at pH of 7. Most body fluids are near neutral, but blood in particular must stay tightly between 7.35 and 7.45. If pH drifts outside that range, cells and enzymes can function properly. That's where buffers come in. These are chemical systems that resist drastic changes in pH. The key example is the carbonic acid bicarbonate buffer in the blood, which helps stabilize pH by absorbing and releasing hydrogen ions as needed. Acids are substances that release hydrogen ions into a solution, increasing the solution's acidity. The more hydrogen ions released, the lower the pH. For example, hydrochloric acid in the stomach helps digest food and kill pathogens. Strong acids dissociate completely in water, releasing many hydrogen ions, while weak acids only partially dissociate, which make them less reactive. Acids are involved in many physiological processes, from digestion to cellular respiration. Bases are the chemical opposite of acids. They either release hydroxide ions or directly bind to hydrogen ions, reducing acidity and increasing pH. For example, sodium hydroxide is a strong base that releases hydroxide ions, while bicarbonate acts as a weak base that binds excess hydrogen ions in the blood. Bases are just as important as acids in the body. They help neutralize acids, stabilize the pH of bodily fluids, and ensure optimal conditions for enzymes and cells to function properly. An imbalance in base levels, like alkalosis, can be just as harmful as too much acidity. The pH scale is a way to measure the concentration of hydrogen ions in solution. It ranges from 0 to 14, with 7 being neutral. Values below 7 are acidic, meaning higher hydrogen concentrations, while values above 7 are basic or alkaline. Importantly, the pH scale is logarithmic. Each number represents a 10-fold difference, so a solution with a pH of 5 is 10 times more acidic than one with a pH of 6. The body keeps blood pH within a very narrow range, 7.35 to 7.45, because even small changes can disrupt enzyme function and lead to dangerous conditions like acidosis or alkalosis. Buffers are chemical systems that help maintain a stable pH by reacting with excess hydrogen or hydroxide ions. When an acid or base is introduced to a system, buffers work to minimize the impact. One of the most important buffers in the body is the carbonic acid bicarbonate buffer system in the blood. When blood becomes too acidic, bicarbonate ions with hydrogen ions binds to hydrogen ions to form carbonic acid. When it's too basic, carbonic acid dissociates to release more hydrogen ions. These reversible reactions help maintain homeostasis. Without buffers, even more minor changes to metabolism could lead to dangerous shifts in pH. Carbohydrates are one of the body's main source of energy. They're made up of carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. The simplest form are monosaccharides, which include glucose, the main fuel source for cellular respiration. Disaccharides are pairs of sugars like sucrose, which is table sugar. Polysaccharides are long chains and include glycogen, which we store in our liver and muscles for quick energy use. Starch and cellulose are plant based polysaccharides. Starch can be digested, cellulose cannot. When we eat carbs, enzymes break them down into monosaccharides, especially glucose, which is then used to produce ATP, the energy molecule all cells rely on. Monosaccharides are the simplest form of carbohydrates. They consist of just one sugar unit, making them easy for the body to absorb and use for immediate energy. Glucose is the most important monosaccharide. It fuels nearly every cell and is essential for ATP production through cellular respiration. Fructose and galactose are more also common and often converted into glucose by the liver. These sugars are found in fruits, honeys, and milk. Because monosaccharides are small, they're rapidly transported into cells for quick energy or stored for later use. Disaccharides are made by linking two monosaccharides together through dehydration synthesis. A water molecule is removed in the process. To digest them, enzymes break them down via hydrolysis. Sucrose or table sugar is the most familiar disaccharide. Lactose, the sugar in milk, and maltose, found in germinating grains, and others. These sugars can't be absorbed in disaccharide form, so the digestive system breaks them into monosaccharides first. Lactose intolerance, for instance, occurs when the body's lack of enzymes needs to split lactose into glucose and galactose. Polysaccharides are long, complex carbohydrates composed of many monosaccharides linked together. In human, the most important polysaccharide is glycogen, which serves as a form of energy storage in the liver and muscles. When blood glucose levels drop, glycogen is broken down into glucose. In your diet, we also consume starch and cellulose from plants. Starch is digestible and provides a major energy source. Cellulose, on the other hand, cannot be digested by human enzymes. It plays an important role as dietary fiber, helping maintain bowel health and regularity. Glucose is the most important sugar in the human body. It powers nearly every cell through a process called cellular respiration, which generates ATP. After we eat, excess glucose is stored as glycogen in the liver and muscles. When the body needs energy between meals or during exercise, glycogen is broken down back into glucose. This regulation is tightly controlled by hormones. Insulin helps lower blood glucose by promoting its uptake into the cells, while glucagon does the opposite. It signals the liver to release glucose from glycogen stores. This balance is crucial for maintaining stable energy levels. Lipids are a diverse group of hydrophobic molecules that play many vital roles in the body. Unlike carbohydrates, lipids do not mix well with water due to their nonpolar nature. Triglycerides are the body's main long-term energy storage molecule. They store more energy per gram than carbs. Phospholipids form the bilayer of every cell membrane, helping regulate what enters and leaves the cell. Steroids like cholesterol and sex hormones are lipids with a ring structure involved in signaling and membrane fluidity. Fatty acids can be saturated or unsaturated, determine the physical properties of the fats. Lipids also cushion organs and provide insulation, especially in the skin and around vital structures. Triglycerides are most common type of lipids in the body and serve as our primary energy reservoir. Each triglyceride molecule consists of one glycerol backbone bound to three fatty acid chains. These are stored in adipose tissue and can be broken down into release energy during fasting or exercise. Triglycerides also help insulate the body and cushion internal organs from mechanical shock. The type of fatty acids, saturated or unsaturated, affects whether the fat is solid or liquid at room temperature, which has implications for health and metabolism. Phospholipids are essential for life because they make up the structures of all cell membranes. Each molecule contains a hydrophilic or water attracting and phosphate head and two hydrophobic or water repelling fatty acid tails. When placed in water, they naturally arrange into bilayers, with heads facing out towards water and tails hidden inside. This unique structure forms a flexible yet protective barrier that controls what enters and exits the cell. Membrane proteins and cholesterols are embedded within this bilayer, working with phospholipids to maintain membrane function and fluidity. Steroids are lipids characterized by a four-ring structure and are crucial for both structural and regulatory roles in the body. Cholesterol is most familiar steroid. It stabilizes the phospholipid bilayer in cell membranes and is the precursor for other steroid molecules. From cholesterol, the body produces sex hormones like estrogen and testosterone, which regulate reproductive systems and secondary sex characteristics. Corticosteroids like cortisol help control metabolism, stress response, and immune activity. Although steroids are lipids, they differ significantly from fats in both structure and function. Fatty acids are the building blocks of triglycerides and phospholipids. The difference between saturated and unsaturated fats lies in their chemical structure. Saturated fatty acids have no double bonded between carbon atoms, making them straight and tightly packed. This is why they are solid at room temperature, like butter. Unsaturated fats have only one or more double bonds that introduce kinks in the chain, preventing tightly packed and making them liquid like plant oils. The type of fat consumed in the diet affects heart health, cholesterol levels, and inflammation. Proteins are the most diverse and functionally important macromolecules in the body. They're made up of amino acids linked by peptide bonds and folded into complex shapes that determine their specificity. Proteins serve structural roles like forming connective tissues, act as hormones and antibiotics, and transport substances like oxygen. One of the most important classes of proteins is enzymes. These act as biological catalysts that dramatically increase the speed of a chemical reaction by lowering the activation energy required. Enzymes are highly specific to their substrates and remain unchanged after the reaction. Without them, most of the body's chemical processes would occur too slowly to support life. Protein function depends entirely on the structure. At the simplest level, the primary structure is the specific sequence of amino acids in the chain. This sequence folds into secondary structures like alpha helices or beta sheets, held together by hydrogen bonds. As the protein structure continues folding into a more complex 3D shape, it reaches its tertiary structure. Some proteins have quaternary structures, where multiple chains come together to form a functional unit, as in hemoglobin. If this structure is disrupted by changes in pH, temperature, or chemicals, the protein becomes denatured and loses its function. That's why maintaining homeostasis is so important for protein stability. Enzymes are specialized proteins that accelerate chemical reactions by lowering the activation energy needed to get enzymes started. Each enzyme is highly specific to its substrate. Think of it like a key fitting into a lock. Once the reaction occurs, the enzyme is free to catalyze another round, making it very efficient. Enzyme activity is sensitive to environmental conditions. If the pH or temperature is outside the enzyme's optimal range, it may denature and stop functioning. Enzymes play essential roles in digestion, metabolism, DNA replication, and virtually every other physiological process. Nucleic acids are macromolecules that store and transmit genetic information. They're made up of repeating units called nucleotides, each composed of a sugar phosphate group and a nitrogenous base. DNA holds the instructions for building proteins and is found in the nucleus. RNA helps carry out those instructions during protein synthesis. DNA uses the bases adenine, thymine, cytosine, and guanine, while RNA replaces thymine with uracil. Another critical nucleotide-based molecule is ATP, adenosine triphosphate. ATP stores energy in its highly energy phosphate bonds. When a bond is broken, energy is released and used to power nearly every cellular process from muscle contractions to nerve signaling. Nucleotides are the building blocks of all nucleic acids. Each nucleotide includes a sugar, either ribose or deoxyribose, a phosphate group, and one of the 5 nitrogenous bases, adenine, thymine, cytosine, guanine, or uracil. These nucleotides link together in long chains to form DNA or RNA, storing and transmitting genetic instructions. But the nucleotides also serve other purposes. ATP, for example, is a single nucleotide that stores energy. So while nucleotides are best for genetics, they're also crucial for powering cellular functions. DNA and RNA are the 2 types of nucleic acids, each with its own role. DNA holds the body's genetic code. It's like a master blueprint stored in the nucleus. It has a double helix structure and base pairs, adenine with thymine and cytosine and guanine, forming the rungs of the ladder. RNA, on the other hand, is single-stranded and helps carry out instructions stored in DNA. It replaces thymine with uracil and is active in the cytoplasm, where it helps assemble proteins. Both DNA and RNA are essential. Without them, cells wouldn't be able to grow, divide, or repair themselves. ATP, or adenosine triphosphate, is often called the energy currency of the cell. It's a modified nucleotide made up of adenine, ribose sugar, and 3 phosphate groups. The bond between these phosphates stores potential energy. When a cell needs energy, it breaks one of these bonds in a reaction called hydrolysis, converting ATP to ADP and releasing energy. This energy powers virtually everything in the body: muscle contractions, active transport of substances across membranes, nerve impulses, and the synthesis of macromolecules. Without ATP, cells wouldn't be able to function. In summary, this chapter gives us the chemical foundation for understanding the human body. Everything from muscle movement to hormone signaling begins at the atomic and molecular levels. Atoms form molecules through chemical bonds, and those molecules, especially organic ones like carbs, lipids, proteins, and nucleic acids, carry out the work of the cell. Water's unique protein structure supports nearly all biological processes and maintains the right pH through acids, bases, and buffers is critical for function. Enzymes ensure the reactions happen fast enough to sustain life, while ATP provides the energy to make it all happen. Mastering these concepts will help us make sense of every physiological system moving forward.